Genetically engineered insulin. This is biotechnological application in medicine part 2. What is insulin? Insulin is a hormone which controls blood glucose level by converting excess amount of glucose to galactose. In absence or lack of insulin, a human being, develops disease called diabetes. And so importance controls blood glucose level, and in the absence of or lack of insulin, a person develops diabetes. Insulin is produced by beta cells of Langerhans of small intestine. Now, why genetically engineered insulin is preferred over non-human insulin? Because of mainly two reasons. Genetically engineered insulin can be produced in large scale in short period of time using E. coli as host cell, means as using recombinant DNA technology. And genetically engineered insulin is identical to the human insulin because the same DNA molecule is inserted in E. coli cells. So they do not produce any immune response, the example allergy or any other type of response against when that insulin is inserted or insulin is used as medicine in diabetic patients. Whereas in case of non-human insulin extracted from slaughtered cattle or pig, liver of slaughtered cattle or pig, immune responses like allergy is very common. Now coming to maturation of insulin. How insulin is produced how insulin is maturated in human body. In human body, insulin is produced in proactive or inactive form called prohormone or proinsulin by beta cells of Langerhans. It is made up of A and B polypeptide or peptide chain linked together by disulfide bond in the active form, whereas the inactive form contains one additional C chain. So, when insulin is required in our body, for example, when blood glucose level rises, then what happens? Our body removes the C chain from the insulin so that the inactive proinsulin gets converted to active insulin. Now coming to the next point, that what was the main challenge in production of genetically engineered insulin? The main challenge was to produce genetically engineered insulin in mature form. Till now we studied that the mature form contains only A and B chain linked together by disulfide bond, whereas it is produced in proinsulin form. So to produce or to add these two chains, A and B chain, was difficult. The scientists found it difficult to make or prepare insulin as it contains two peptide chain. Now, how the problem was solved? In 1983, an American company, Eli Lilly, found the solution. So what was the solution? Following are the steps. Two separate DNA strands, synthesizing A and B peptide chain, are artificially constructed. First, artificial construction of two DNA strands, two separate strands synthesizing A and B peptide chain respectively. Then introducing those two strands separately in E. coli cells carrying plasmids. I repeat, A chain and B chain are introduced in the plasmids present in E. coli cells separately. Then replication of the E. coli cells took place in and A and B chain along with the bacterial cells replicated or multiplied. The amount increased in separate bacterial cells. Then A and B chain are extracted separately. Then they are joined together by forming disulfide bond to construct insulin. All of this is done in the last step is done in vitro, that is in laboratory. It did not occur inside human being or inside E. coli cells. Now coming to the next step, that is molecular diagnosis. That is another method of biological application of in medicine. So what are the methods used? Basically 3 methods are used: PCR, ELISA and recombinant DNA technology. We'll discuss this later. First, one question: why molecular diagnosis is preferred over conventional methods of diagnosis? In case of conventional methods of diagnosis, identification of a disease can only be possible once the symptoms of the disease is found or is observed. Once the symptoms are developed, then only identification of disease is possible. But at this time, the concentration of the pathogen increases. So the disease develops and the treatment becomes very difficult. But in case of molecular diagnosis, identification can be done when the pathogen concentration is very low. So we can also say before the development of the disease or during the early stage of a disease. So the treatment also becomes easier. Now coming to roles of each method. PCR is polymerase chain reaction. It can amplify any DNA fragment which may be the reason behind the disease. For example, in case of patient who may develop AIDS, for example HIV infected person or the suspected person who can develop HIV infection, from those patients, DNA, very small amount of DNA is extracted, then it is amplified by using PCR, then further diagnosis is done, because it is easier if the suspected DNA fragment has the source of suspected DNA fragments is more. If it's very less, then it won't be available for testing or diagnosis. Now coming to the next one, that is enzyme linked immunosorbent assay, in short ELISA. I repeat, enzyme linked immunosorbent assay, ELISA. We all know that antigens are the chemicals or the proteins produced by the pathogen, and our body produces antibodies against a specific antigen. So this method is used in ELISA. The antigens produced by a pathogen or the antibodies which are synthesized or created against those antibodies are detected using ELISA. This technique help us in detection of HIV or other complex diseases in very early stage, as antigens are produced by the pathogens as well as our body also produces antibodies. Now coming to next one, that is recombinant DNA technology. In case of recombinant DNA technology, first the steps are like this. Single-stranded DNA or RNA is attached with radioactive molecule that is called probe. Then radio-labeled RNA or DNA strand is allowed to hybridize. Hybrid is a molecule which consists of both different, it's better to say different types of genetic material. Suppose, for example, DNA will form or DNA will bind with RNA, or RNA will bind with DNA, vice versa. Hybridize a molecule, double-stranded DNA molecule. The next step, if this complex contain mutated gene or genes, then it cannot be seen under autoradiograph or using autoradiography, whereas non-mutated hybrid will be observed using autoradiography. It is a technique to detect. Autoradiography is a technique to detect radioactive molecules. So in this session we discussed about genetically engineered insulin and molecular diagnosis. In case of genetically engineered, we discussed what is insulin, importance of insulin, why GE insulin, that is genetically engineered insulin is preferred over non-human insulin. Then maturation of insulin. Here maturation of insulin. The next point discussion is done. What was the main challenge in GE insulin production? How the problem was solved? Then coming to the molecular diagnosis. Three methods: PCR, ELISA, recombinant DNA technology, and why molecular diagnosis is better or more useful than conventional methods of diagnosis. Then how PCR, ELISA and recombinant DNA technology is used. That also discussed.